Hey everyone, it's Mr. Wolf here, and in this video I'm going to be discussing the question, what does the building and the breaking of molecules in organisms? And the short answer to this is enzymes. Enzymes carry out these processes, but in this video let's talk about how that actually goes down. So before we talk about enzymes, let's just talk about any type of reaction. So this graph may look complicated, but it's really not too bad. On the x-axis, we have the progress of a reaction. So for any reaction to occur, we could be talking about molecules breaking down or molecules being built. And on the y-axis, we have energy. And you'll notice that in order to get from our reactants to the products, there's this hump or hill that we have to get over in order for this to happen. So it's almost like we need an input of energy. This input of energy is going to be called our activation energy. It's the amount of energy required to start a chemical reaction. And you'll notice that this black line would just be what would it normally require for a reaction to take place, but then we have this red line. And when we have this red line, that's when we would have something like a catalyst present. And so a catalyst is just anything that speeds up chemical reactions. And they work by lowering this activation energy. So notice that we just have a smaller hill to get over in terms of the energy that we need in order for this reaction to occur. And because of that, we're going to have a lot more reactions happening a whole lot faster. All right, so how do enzymes come into play? Well, when you have a catalyst, so something that speeds up a chemical reaction, and it's a protein that's doing it, that's called an enzyme. So in other words, enzymes are a type of protein that speed up chemical reactions. And here's kind of an example of what that protein would look like. And each little dot right here would be an atom that makes up this protein, this molecule. Right. So how do enzymes speed up reactions that break down molecules? Well, we'll first start with our enzyme right here. That's our black shape, right? And then what we need is our reactants in any type of reaction. And when we're talking about enzymes, when we say reactants, we might also say substrates. So the word substrate is literally just another word for reactant. So let's say we have this polysaccharide reactant, our polysaccharide substrate, and we also have our enzyme present. What's going to happen is you'll notice that this enzyme has this part of it. This is called the active site, where my polysaccharide could kind of perfectly attach right here. And because of that, my substrate is going to bind to the active site of my enzyme. And once this happens, the enzyme is actually going to change its shape a little bit, and you'll notice that that's going to rip my polysaccharide apart, and it's going to create my products. Now, one important thing to notice here is that after this reaction takes place, my enzyme looks exactly like it did before, which means that it could continue to do the reaction again and again. So you could think of enzymes like the Energizer Bunny. Enzymes can be reused again and again. As long as I have more substrates, this enzyme could continue doing the same exact reaction over and over and over again. So here's another important part about enzymes, is that they are super specific. And you could get that from just looking at how this enzyme almost perfectly matches up with our substrate. So not only do they match up with specific substrates, but they only do one type of reaction for one type of molecule. So let's look at some examples for this. So the enzyme to the right only breaks down a polysaccharide. The polysaccharide fits in and it'll break it apart. So that means if we had, let's say, a protein polymer, since we're modeling it as circles in this case, these circles wouldn't perfectly fit in the active site, and therefore this enzyme couldn't break up an amino acid. There would have to be another specific enzyme that does that. Now, what if you have carbohydrates, but they're just monomers? Can you build them up? No. So even though it may look like that this enzyme might be able to fit our monosaccharides right here, this enzyme only breaks down polysaccharides. It only breaks down carbohydrate polymers. You would have to have another type of enzyme that builds up a polysaccharide. So let's look at another example of how enzymes work. In this case, we're going to be talking about enzymes building molecules. And you're going to see a lot of similarities to what we had before. So for instance, we're going to start with our substrates. Remember, those are just our reactants. And in this case, we have two amino acids. And in order to bring these amino acids together to speed up this process, we have an enzyme. And again, an enzyme, even though we're modeling it as this just kind of black shape right here, remember this is actually what an enzyme would look like. All right, so this enzyme for this protein has a different active site. It's specific for my 2 amino acids to bind right here. And that's what they do. And once that happens, this enzyme is going to change its shape in a little bit in some way that actually allows these amino acids to bind together. And so my amino acid is going to create my products. They're going to get released. And before I had amino acids, now I have the beginning of a protein right here. And again, another important part is that this enzyme has the exact same shape as it did at the very beginning, which means that enzymes are like the Energizer Bunny. They could be reused again, again, and again. So if we had more amino acids around, this protein could continue to build and build and build and build.